Guys, welcome back to the channel. So in this class, we'll talk about antibodies and their structure and their function. We'll also talk about primary and secondary responses. This will be a direct continuation of the last class, which we talked about lymphocytes and B cells. And we know that B cells secrete antibodies. So this class will go a bit more deeper about their structure. So let's begin. So let's just have a look at what the structure of antibody is first. It is at the bottom right. So it's like this Y-shaped structure. It is made of globular glycoproteins, and we say that it has a quaternary structure. And if you can recall from the proteins chapter, the biological molecules chapter under proteins, we know that a quaternary structure means it consists of two or more polypeptides in the entire protein structure. So it's made of 4 polypeptides, 2 long or heavy, and 2 short or light chains. So these long ones over here, this is a long or heavy chain. And the small one here, that's a short or light chain. So they're held together by disulfide bonds. So there's multiple disulfide bonds. First one you talk about is the one that holds the long chains together. And there's also one which holds a long and short chain on both sides together. So when they're held together, it forms variable regions. So variable regions are four of them exactly. And the 4 variable regions together, they form something called the antigen binding site. So it's called a variable region because it differs for antibodies, because each antibody will have a different variable region, and if it has different variable regions, they will form different antigen binding sites, because one antibody can only be complementary to one type of antigen. So the sequences of the amino acids in the antigen binding site, which is over here, or part of the variable region, they contribute to the specific 3D shape of the antibody. So it means that this specificity only allows you to bind to one type of antibody. Only allows it to bind to a specific type of antigen. So the antigen binding site form the variable region because it differs on every antibody molecule, like I just said. So the hinge region, which is over here between the 2 long chains, this gives flexibility to the antibody molecule to bind around antigens. This flexibility allows it to maintain the structure of the antibody while being able to bind around antigens. There's also the strong disulfide bonds. So these bonds are very strong, as we talked about them. When they're very strong, it requires a lot of energy to disrupt the entire structure of the antibody. So this helps to maintain the quaternary structure as well. So, sorry. Okay, my bad. So next we'll talk about what exactly do antibodies do in the body. So the first thing is the special types of antibodies, which are known as antitoxins, because some pathogens secrete toxins, and when these antitoxins bind to the toxins, it makes them harmless. So let's look at this picture here, the first column here. So the virus, for example, secretes some type of toxin, and this antibody can bind with the bacterial toxins and prevent them from damaging cells. So look at over here. So this binds to the toxins, preventing them from doing anything or having any effect. So when they bind to it, it prevents them from entering host cells and damaging them. And they do this same with viruses. So when 2 of them, as shown in the diagram, so basically we just take it as the antibody binds to the virus, which prevents them from entering host cells and causing any damage. Furthermore, some antibodies, they have multiple binding sites. So this causes agglutination. So agglutination is basically when a large number of things clump up together. So over here, multiple antigen binding sites basically mean that, for example, it has two regions, so on the front and the back, which both of them can bind to a pathogen or the antigen, sorry. So over here, this binds to this binds to this, and these are all clumped up together. So when this happens, when agglutination happens, so the bacteria are limited to a specific region in your body. So it prevents them from spreading, prevents them from going to other regions and dividing and spreading there as well. So antibodies can also coat the bacteria, which also help to make them easier to be recognized by neutrophils, because neutrophils have those antibody receptors. So as you talked about that in how neutrophils actually bind to the bacteria itself. So first of all, antibodies will coat the bacteria. And phagocytes have receptors for the heavy polypeptide chain of antibodies. So when they bind to this, they recognize, oh, it's a phagocyte, or sorry, oh, it's a pathogen. So I need to engulf this. So that's one function. They can also bind to the flagella. Flagella is that organelle which causes a beating movement, causing the pathogen to spread or move around. So when they attach to the flagella, it makes them less effective. So they cannot beat as fast, or pathogens cannot move as fast. So it makes them easier for, first of all, phagocytes to engulf them. And also it reduces the spread of around the body. So it does not travel as fast and start dividing and get away from the white blood cells. So the next thing is also about antibodies working with other molecules to punch holes in cells. So over here, when they work with other molecules, they punch holes in cells. So what happens is water will enter, and since the membrane is weakened, eventually what will happen is that a lot of pressure will build up, causing the pathogen to burst. And I made a mistake here, so with point 1. So what my mistake was that antibodies they can combine with viruses and bacterial toxins, which prevents them from entering or damaging cells. And the one I was talking about antitoxins, that was my bad. So what happens is that they neutralize the toxins, which make them completely harmless. So they have no more effect. So sorry for the mix-up. So next part about antibodies is about primary and secondary responses. So primary response is when antigen enters the body for the first time, and secondary response is when the same antigen enters the second time. So obviously the primary response is much slower, because when the antigen first enters, it needs to undergo clonal selection and expansion, and eventually it'll go up, and then this is the concentration of the antibody is building up as the number of B cells increase, and then eventually it'll decrease. So example over here, there's a massive spike in the antibody concentration, and the antibody concentration of the secondary response is much greater than the first response. So what happens is after the primary response is immunological memory, because memory cells are already formed as a result of primary response. So the time taken for the antibodies to the concentration to increase is shorter, and the concentration is greater, and the antibodies not disappear completely, but they circulate in the body for a while. But for primary response, they eventually are destroyed or broken down by the body. So I hope that helps. So this will conclude this lesson about antibody structure and function, and about primary and secondary response. So we'll wrap up this chapter in the next video. As usual, I hope you find these videos helpful. If you do find them helpful, you could consider liking and subscribing and sharing them with your friends. If you have any questions, you could leave them in the comment section. Till the next video, thank you. All the best for your study.